We are going to, the next lecture will be the last endocrine lecture. Your test is scheduled, I think, for 2 weeks from today, right? Not next week, but the week after. Blood is usually like a lecture and a half. We may start on it next time, which means we'll, as usual, start the new material like one lecture before the test, which is how it's kind of been going. I guess it's not that bad because it gives you an extra weekend to study. You know, we could have the test a week from Wednesday, but you'll have an extra weekend to study. So yeah, that's it. So the last organ that we have to do, the only one that we didn't do last time, is the ovaries. And then pretty much we are done with, there's no new hormones after that. There's a couple that I refer to in here from other endocrine organs, but I don't test off over those. I just put them in there because in the future, if, for instance, you go into pathophysiology, a lot of people take their endocrine table with them. So then all they have to do is add new disorders, right? They already know like the function and the targets and everything, or in other classes they use those. So it's going to give you a place to put those for the future, but I won't test over them yet. Okay, so the ovaries. We already know most of the ovary story because it's almost the same story as the testes story because basically they are the same organs. Of course, the testes, you're doing two primary functions. One of them is secretion of androgens, and the other one is spermatogenesis. So with the ovaries, you're going to do two primary functions as well. Secretion of estrogens and oogenesis. But then in addition, the ovaries have an extra function because, of course, females have an extra function, which is the possibility of housing a pregnancy, right? So in addition to those two functions, the ovaries are also going to tell the uterus what to do. Okay, so I don't know how much of this you did in anatomy, so I'm just going to give you like this little preview of like ovarian function for just a second. So with the ovaries, what you're going to do is every single 28-ish day cycle, one of these little guys is called a follicle, and if you stimulate it, follicle stimulating hormone, then it will start to go through the process of meiosis, right? Okay, and generally with human females, you're going to do one of these per month. And what's going to happen is this starts out with 46 chromosomes, but in order for it to be viable, right, you're going to have to decrease to 23 chromosomes, right? And then whatever you ovulate and gets fertilized will go back to 46 chromosomes, right? Because the sperm has 46 or 23 and the egg has 23. So this process is going to go through meiosis and increasing the size of this big follicle and then ovulation, right? Okay, so this is generally going to happen in the ovarian cortex. The medulla of the ovary doesn't really do anything special. It's generally where the blood vessels come and go from and stuff like that. So that's sort of your story of the ovaries. Now, I'm going to do a simplified version of ovarian function because just so you know, when I took physiology, I think we did 2.5 weeks on the female reproductive cycle. But if I did that, I couldn't have taught you the other 30 hormones, right? So I'm going to do a simplified version of it. Okay, so what do the estrogens do? The estrogens are, of course, secreted from the ovaries. And the estrogens, remember our story that we keep telling? The estrogens right here, these are collectively considered the estrogens. The most common one that people refer to as estradiol. These are actually derived from androgens because remember these were the androgens. And what happens is in women you have a lot more of these enzymes that can do the conversion of androgens to estrogens. Okay, does that make sense? So what happens, and we will see, is you actually make androgens and then you do conversion of them if you are a functional female. So we know testosterone's functions, right? Testosterone, of course, feeds back to the brain, right? But in addition, it also causes bone mass growth and skeletal growth and axillary hair growth and facial hair growth and changes the voice and causes the development of the secondary sex characteristics in males. Estrogens do something similar. Estrogens are responsible for both development of the primary sex characteristics in females. That means your parts are all in the right place and functional. Now, I told you this before, the default anatomic gender in humans is female, but she's not going to be functional unless she gets a shot of estrogen to make the ovaries and everything function properly. So she's going to look female, but if you don't get a shot of estrogen at the right point in development, you're not going to be functionally female, reproductively functionally, okay? So estrogens do primary sex characteristics, and then pretty much no estrogen to speak of until you hit puberty. So then at puberty, you get the development of the secondary sex characteristics. And the secondary sex characteristics are, of course, the continued development of the reproductive tract and breast development and changes in body fat deposition, and it feeds back to the brain and it does all that stuff, right? That makes your teenage girl act like a teenage girl as she's going through puberty. And mine is going through that right now. She's 14, and I realize how much I failed as a parent when the other day she told me, yes, that she does not believe that dinosaurs are real. I was like, what? She thinks that it's all like an elaborate hoax and dinosaurs aren't real, and she also doesn't believe in evolution, and she's not going to college. She wants to be a hairdresser. I was like, how much could I have possibly failed? I think this is like basically rebellion, don't you? I think she just figured out everything that would bug me, and she's like going to say them all along. I was like, you don't believe that dinosaurs are real? And she's like, no, I think somebody just planted that there, and it's really a cow. I was like, have you seen a Triceratops skull? This is a weird-looking cow. So, yes, I failed. So, female development and female secondary sex characteristics. Here's the whammy, though. Remember how testosterone was responsible for sex drive? Estrogens make you fertile, but they have nothing to do with sex drive. Absolutely nothing to do with sex drive. So you can get knocked up without ever having wanted to have sex, right? And I would like to talk to the management, wouldn't you? About that? I want to talk to the management. Okay, so they are also necessary for the female reproductive cycle, including oogenesis, and estrogens are actually converted androgens. So we are going to look at this story right here. I'm going to simplify it a little bit for you. But we're going to build it. But this time, I think you guys are ready to build it yourselves, aren't you? We already did the testosterone and spermatogenesis story, so I think you guys can do the estrogen and oogenesis story, right? So if I'm going to try to end up secreting estrogen and making some eggs, where does the story start? Yeah, it always starts with the hypothalamus. And relevant hormone from the hypothalamus to start the story is what? Yeah, GnRH. Note to self, I don't know if I've said this to you recently, but if you write GnRH on a test, you have to spell it out the first time you write anything. And the reason is that if you spell out gonadotropin releasing hormone and you get a letter wrong, I can give it to you because I know what you mean. But if you put GnPH, that's wrong. Do you see what I mean? Or GnHR. So I make you spell everything out the first time, and then you can abbreviate it from the rest of the time on. So GnRH goes where? Anterior pituitary. And I want the relevant hormone from the anterior pituitary to cause estrogen, ultimately. What's the relevant hormone? Yeah. Which stands for what? Luteinizing hormone. And luteinizing hormone is going to go to the ovaries. Specific cells in the ovaries, I'm not going to have you learn them because, look, it's actually kind of complicated. LH actually goes to the theca cells, which then causes androgen secretion, and then you actually modify them in different cells. So I'm not going to force you to do that, but let me just show you how that's related to this story really quickly. So please note that the thing that is supporting the developing egg is called a follicle, and follicle-stimulating hormone will become relevant there. And then note that around the follicle are two different kinds of cells, theca and granulosa. So what's generally going to happen, and I don't test on this, but just so you know the story, is that you produce androgens here and they are converted to estrogens here. Okay, so that's just the part of the story that we end up telling. So you can just leave it at ovaries for right here. So what's going to happen is ultimately the ovaries are going to make androgens and then convert them into estrogens. But we're just going to skip that part of the story and say that we end up with some estrogens. And then what can you tell me about the chemistry of the estrogens that will tell you how it feeds back? What do you know about estrogens? They're chemical. Yeah, they're steroids. So where are they going to feed back? They feed back to where? They feed all the way back to the hypothalamus because they are lipid soluble, and they also feed back to the anterior pituitary, right? So, of course, this is way more cyclical than the androgen secretion in males, right? Males secrete pretty regular amounts of androgens over long periods of time, but remember your estrogen secretion goes up and down over about a 28-day cycle, okay? We're not going to make you memorize the cycle, but it does. It's pretty dramatically different at different points in the cycle. So we've got our estrogens and estrogen secretion, but now we need to take care of the other function of the ovaries, which is oogenesis, right? Oogenesis, which is the production of a haploid egg and then eventual ovulation. So the anterior pituitary is going to make what relevant hormone that will result in oogenesis? FSH, which is follicle stimulating hormone. And FSH is going to go to the ovaries. It goes to different cells for the ovaries. I don't usually test on them because I'm oversimplifying it. And then it's going to result in oogenesis. But same story that we told before. Only if you have estrogens, right? Remember that you needed the androgens or testosterone in order to do spermatogenesis. You need the estrogens in order to do oogenesis, okay? So this is going to happen about once every 28-day cycle in which you actually do meiosis and then ovulation, right? Meiosis and then ovulation. So the question is the same as it was before. How does your body know when you have just done oogenesis and ovulation? What do you release? Yeah, you release inhibin. Whereas this would be released pretty frequently with males, it's released about once every cycle with females. And then inhibin feeds back to where? Yeah, it feeds back to the anterior pituitary only, and it only cuts off production of FSH. Go ahead.
So what effect does Clomid have on the brain for you to produce more?
I can't remember what Clomid was. I think that Clomid is an FSH agonist. I think that's what it is. We can look it up. I'm sure it's very easy to understand with this whole story, but I think it's an FSH agonist that causes hyperovulation. Makes you ovulate more often. So if you email me that, I'll look it up and figure out how it fits into the story because it's a good question. Okay, so I just did oogenesis and I made inhibin. So the thing now is that we have to add in the other part of the story, and the other part of the story that makes the female reproductive tract really complicated is, in addition to doing the two functions that we just talked about, the female reproductive tract has to also prepare for the possibility of an implantation, right, which would be a pregnancy, and then maintain the pregnancy over the course of however long it takes, 10 months. People say 9 months, but we know better, don't we? Because it's 40 weeks. All right, so what's going to happen is, watch here. So we go through this process of going through meiosis and then ovulation, right? But what happens is when this thing is ovulated, it takes about a week for it to potentially implant in the uterus, okay? So there is a lag time in between ovulation and implantation. Now, so what would happen if a pregnancy was going to occur is you do ovulation and then you have sex at some point during that, and the sperm actually stay in the reproductive system for a few days. And so the fertilization could occur. Let's say the fertilization occurred a couple of days after you ovulated. But the fertilization is not a pregnancy. Physiologically, your body does not know anything occurred when fertilization occurred, because all I had was something that was going through a tube, the fallopian tube, right? And something else was going through a tube, which was a sperm, right? And they meet in the tube. But they actually don't interact directly with the woman's body. So there is no way for her to know that fertilization occurred. So from a physiological perspective, life does not begin at fertilization. Life begins at implantation at the very, very earliest. As a matter of fact, 50% of fertilization events do not implant. So if you've had 10 children, you've probably had 20 fertilization events, for instance. Does that make sense? Okay, so, but what's going to happen is I'm going to have to actually tell the uterus when a possible fertilization event could occur in implantation, right? And the way I tell the uterus is that during this whole process, what is happening is I am actually starting to secrete not just estrogen, but also a new hormone called progesterone. And progesterone is the hormone that you use for the ovaries to talk to the uterus, okay? And what they are going to do is, as the progesterone gets higher, it is progestation. What do you think it's going to tell the uterus? What do you think it tells? It to get ready. What it's going to tell is it's going to tell the uterus to get ready. And how does the uterus get ready? By increasing the thickness of the lining of the uterus. So what's going to happen is the ovaries are going to secrete progesterone, and the levels of progesterone are going to go up from here, and then they have to stay high after ovulation, right? Because it's after ovulation when the implantation could occur. So this little supporting structure that was around the developing oocyte, this thing actually stays behind and continues to secrete progesterone for about a week. 10 days-ish after ovulation. And so what happens is it's going to continue to secrete progesterone and continue to secrete progesterone, and it's basically making sure that you have this nice window in which there is an egg potentially, and the thickened wall of the uterus is prepared for an implantation. And then after you have gotten out of the window, in which an implantation really is highly unlikely, then your levels of progesterone drop, right? And then you start this whole process over again in the uterus. But in the ovary, what's happening in the uterus? When your progesterone level drops. Yeah, you menstruate, you slough it off, right? And then you start the whole process again. So the part of the reason that this is really complicated is it really has to be carefully timed in a woman. And also that in addition to doing your 2 typical functions, you also have to tell the uterus what's happening, right? Okay, so you need to tell the uterus that an implantation is possible or not possible. Does that make sense? Okay, so progesterone is progestation. Now, let's say the woman did not get pregnant. If the woman did not get pregnant, no implantation occurred, then the progesterone level will drop. If, however, an implantation did occur, what's going to happen to the progesterone level? We hope. Yeah, it should stay high throughout the pregnancy, right? It's progestation, right? As a matter of fact, if your progesterone level drops in the early stages of pregnancy, a lot of times it causes miscarriage. Okay, all right, so progesterone maintains the lining of the uterus in preparation for a pregnancy or until no implantation occurs. This is pregnancy supporting. So the ovaries also produce inhibin. We've got that one. It's a peptide. And then one more that they only produce during childbirth called relaxin. This one's kind of an interesting one. Relaxin loosens up the connective tissue in the pelvis. But since it goes into the bloodstream, it doesn't just loosen up the connective tissue in the pelvis. It loosens up the connective tissue everywhere. And I didn't go into labor. I have a heart problem, so I didn't have this experience. But a lot of women report that when relaxin starts coursing through your veins, you can feel it because it feels like if you threw a ball, your arm would go with it, and you're just all floppy. So it actually loosens everything up. It also relaxes some of the smooth muscle, although not in your uterus because you wouldn't want it relaxed in your uterus, right? You actually need that one to contract. So that only happens during childbirth. Okay, so the question is here. Can you reconstruct the chain of hormone secretion that begins in the hypothalamus and ends with oogenesis and the production of estrogen? Yes, you can, right? Okay, so clinical connections, just so that you can properly argue with the people that you disagree with. Let's talk about this drug that is controversial in some circles. RU486, and there's a bunch of different ones that are similar to this, but I'm going to talk about this one. This one is also called mifepristone. And this one, depending on your political leanings, you might refer to it as the abortion pill or the morning after pill. But I want to talk about where it works and when it works and how it's related to this story, right? Because you need to understand the physiology before you have your opinion. A lot of people decide their opinion before they actually know the facts. It's nice to do it the other way. So this one is a progesterone antagonist, okay? So what it is going to do is it is going to block the effect of progesterone on the uterus, okay? And it's really, really supposed to be taken during about 72 hours after, let's go slightly less controversial, and it's interesting that it's less controversial to talk about a sexual assault, but it is less controversial to talk about a sexual assault. So if a woman has a sexual assault and she goes into the emergency room, she can choose to have this treatment, among other treatments. And what would happen is she would be given a series of pills, and the first one would be given in the hospital, and it's progesterone antagonist. And what's going to happen is if she was sexually assaulted, she's not pregnant yet. Do you agree? Because pregnancy occurs up to a week after sex, right? Fertilization could have occurred, but the implantation did not occur. And so at that time, in that window between fertilization and implantation, first of all, you don't even know if fertilization occurred because there's no way for your body to tell you. But between fertilization and implantation, if you give a progesterone antagonist, what's going to happen? Yeah, you are going to slough off the lining of the uterus so no implantation could occur. So from a physiological perspective, this is a pregnancy preventer, okay? Because pregnancy occurs at implantation, right? Now, part of the reason that it's a little controversial is that if it occurs, if you give this in the very, very early stages, like a couple of days after implantation, then sometimes it can actually cause you to slough off an implantation. It's not effective at doing so, and it's not very safe for the woman to use. You're supposed to use it within about 72 hours, like pre-implantation. But if it was, a few days forward, then implantation might have occurred, and it could still slough off the lining. It's more painful for the woman. It's not as safe. It's not really a good, effective pregnancy termination mechanism. But that's part of the reason that people really argued, and it's part of the reason that it took a long time before it was actually readily available in Utah, is that people thought of it as a chemical abortion. And some of these are more effective at doing this than others, but most of them are not meant to be used after implantation. Some of them can be, though. So anyway, now you know how you can phrase your argument, regardless of what you believe. Now you can phrase your argument. So this is RU486. It blocks progesterone receptors. Okay, chemical class of hormones, androgens, estrogens, progesterone are all steroids, and all the rest are peptides. Good? Okay, so there's your assignment, and that is the last of the ones that you are going to have to know. I'm going to tell you just about these other ones just because you may run into them in the future, and I want you to at least know that they exist. You are not going to be tested over these unless later on in the semester, for instance, we'll learn a few of them. Like when we do the heart, we're going to learn the one that's released from the heart. But for right now, I'm just going to tell you for kicks and giggles. So some other hormones that may end up being important in your clinical exposure. One of them is called thymosin, that's released from the thymus gland, which is right here. And it is responsible for maturation of T cells, part of your immune system. Okay, that's why they're called T cells, because they mature in the thymus because of thymosin. The pineal gland you probably already know, right? The pineal gland produces melatonin to help you regulate your circadian rhythms. It is controlled by the hypothalamus. The hypothalamus tells it what time it is, and then the pineal gland secretes melatonin in response to it. The digestive tract has a couple of them. This one's kind of interesting. Ghrelin, which is produced by your stomach when you're hungry, which sounds like growling, doesn't it? So this is produced by your stomach, and it causes the hunger. And then there's another one produced by the stomach called gastrin, which causes digestion. And then the heart releases one called ANP, which we will learn later this semester. It's called atrial natriuretic peptide or protein. And it's produced from the atrium of the heart, and it goes to the kidneys, natriuretic, and tells your kidneys when they need to dump more sodium and water. It's produced in response to like prolonged atrial stretching because of maybe high blood pressure, and it drops your blood pressure. The kidney produces EPO, erythropoietin, which is responsible for causing red blood cell synthesis, erythropoiesis. It also produces calcitriol, which is related to vitamin D. And the placenta. So how does your body know when an implantation has occurred? As soon as an implantation has occurred, you start to develop the placenta at the site of implantation, and you start to produce this hormone called HCG, human chorionic gonadotropin, and that is the pregnancy hormone. That is the one that you pee on the stick and is detected when you pee on the stick. But now you and all of your friends are going to know that you don't pee on the stick 2 days after you had sex, because that's just a waste of $20, isn't it? Right? You wait at least a week, and probably 2 weeks would be safer. And then the bigger the placenta, the more HCG is actually produced. It's sometimes a little hard to tell in the very early stages of implantation. So it detects HCG. Interestingly, if you have multiple implantation sites, your HCG is going to go through the roof. That's one of the ways that they can tell that you might have twins, triplets, whatever. Your HCG levels are way, way, way higher with multiple implantation sites. So that's HCG, the pregnancy hormone. And then last but not least, adipose tissue produces this hormone that's been studied a lot lately as a treatment for obesity. It's called leptin. And leptin is your hormone of satiation. It tells you when you're full, tells you when you should stop eating and increase your metabolism. It's the hormone that says, I'm finished taking it in, now we need to burn it. Fascinatingly, some of us produce more leptin, some of us produce it more quickly, and some of us are more sensitive to leptin. And so they are, for instance, looking at it for treatment with obesity. But most of us are not obese because we got full and didn't know we were full. Most of us, like, you get obese just because you're like, this is more fun than not eating, right? And so I'm just going to keep eating and not actually wiggle. So it decreases your appetite and increases your metabolism. One of the things that we do know is that it takes it a little while to go through the bloodstream and to actually cause the response to decrease your appetite. So people tend to eat a lot less if they eat more slowly. It's one of the ways, very good ways of regulating your weight. Is just, they say you should like put down your fork between bites. Who the hell has time for that? I don't even know who has time for that. But it will actually allow your leptin more time to feed back. So anyway, those are the ones. All right, so the last thing to do with endocrine disorders is just categorizing them. This is no new endocrine disorders at all, just the ones that you've already learned. Now you're going to have to categorize them in one of 4 categories. Hyposecretion, hypersecretion, too little, too much, right? Or responsiveness. With responsiveness disorders, there's nothing initially wrong with the level of hormone. It's the target cell response to the hormone. A lot of times the receptor for the hormone is either too sensitive or not sensitive, or there aren't as many receptors or too many receptors, okay? So what we're going to do with these is that we're going to categorize each hormone that we've already done and make sure that we understand how it's related to these terms. But I do want to tell you that this harkens all the way back to the beginning of the semester, because I want to talk about what will happen to an endocrine disorder if you don't treat it for a while. So let's say we've got someone who has all the symptoms of let's say hyperthyroid, okay? So you have like sleeplessness and you've dropped a lot of weight and you've got maybe a little bit of exophthalmos and all of that kind of stuff, trouble with thermoregulation. So if you don't treat it for a while, and you have a hypersecretion disorder for a while, you can end up with a regulation problem, right? If you chronically hypersecrete, do you think you're going to end up with upregulation or downregulation of your receptors? Downregulation. So if you chronically hypersecrete, you can downregulate your receptors, which will cause a what responsiveness problem? Hypo-responsiveness problem. And so do you see, if you've waited for it to get to that point, you're going to have a lot more trouble figuring out the proper treatment. Does that make sense? Because if you, for instance, regulated the level back to what you thought was normal, but your receptors were insensitive, then it's not going to behave the way that you expected it to do. Likewise, what if, for instance, you had an adult male with a hyposecretion of testosterone? Right? So let's say we've got a hyposecretion of testosterone that he doesn't treat for a while for whatever reason. First of all, hyposecretion of testosterone will make you low motivation to go to the doctor, right? It makes you low motivation to do anything and anyone, really, low levels of testosterone. So chronically low levels of testosterone untreated for, let's say, a year, okay? What kind of regulation are you going to get in that situation? You're going to get an upregulation, right? And when you get an upregulation, you're going to get a what kind of responsiveness problem? Hyper-responsiveness, right? Which makes the treatment protocol a little different than you would expect it to be. So you can start out with one and end up with the other, okay? All right, so treatments. Let's just talk clinically about treating endocrine disorders. If you have a hyposecretion, for instance, it's going to be easier to treat than a hypersecretion. Does that make sense to you? Like if you have too little, then you add it back and you try very carefully to get the level right? But if you have too much, then it's harder to treat. Sometimes you need radiation or surgery or antagonists to treat hypersecretion. It's a little harder to treat those. But if you have a hyposecretion, you can add it back. But the chemistry of the hormone will really limit how you can add it back. If it's a peptide-based hormone, you usually have to bypass the digestive system, right? Because if you swallow, for instance, insulin, then you're just going to have an incredibly expensive amino acid supplement, right? You won't actually have functional insulin, right? So if it's a peptide-based hormone, then you are usually limited by your ways of adding it back. Like growth hormone, you have to inject. Insulin, you have to inject. Vasopressin, I thought you had to inject, but she actually has a friend who, or a little boy she takes care of that has vasopressin oral supplements, which I don't understand. But you're going to teach me, right? You're going to go ask the mom and say, how does this work? But generally, peptides, actually, you have to bypass the digestive system. You have to get it in some way other than the digestive system. But with a lipid-soluble hormone, you have so many possibilities of adding it back. If they're lipid-soluble, you can do a patch or a topical cream. So for instance, if you had that low testosterone situation in that male, you can do testosterone creams, you can do testosterone patches, you can do thyroid patches, you can do oral supplementation of thyroid, you can inject it if you need to. It's a really effective way of getting in there really quickly. So with lipid-soluble stuff, you have lots of possibilities for adding it back. So, okay, so now, if we're talking about treating hypersecretion disorder, it depends on why you're hypersecreting. If, for instance, you had a tumor, then you try to get rid of the tumor, right? Surgery, radiation, whatever the hell you need to do. With, for instance, sometimes Graves' disease or hyperthyroid is caused by a thyroid tumor, and they will go in and do surgery to remove the thyroid, and then they'll add radioactive iodine to take out the last of the cells, because iodine like pretty much just goes to the thyroid. So you add radioactive iodine. Sometimes you can try to block the hormone synthesis. It's usually very hard to do that. And if you can't do that, you'll add an antagonist. If you can't take care of the hormone level, you can add an antagonist to actually take care of the target cell response to the hormone. That make sense? Okay, so lastly about disorders is that when you're talking about a disorder, remember that some hormone chains are 3 hormones long, right? So if you start out with the hypothalamus, it's going to secrete a hormone, and then the anterior pituitary responds by secreting another hormone. And then the third endocrine gland responds by secreting another hormone, right? So a classic example of that would be like thyroid, right? Okay, TRH, TSH, T3, and T4. Okay, so what I want to tell you is this, that what manifests as a thyroid disorder, and let's say I've got a person who's hypothyroid. You gain weight, you're really exhausted, all of those symptoms associated with hypothyroid. Do you agree that the problem could be at the thyroid or the anterior pituitary or the hypothalamus? Do you agree? Right? You could have hypothyroid that would manifest as a thyroid disorder that didn't actually occur in the thyroid. Does that make sense? So the question is, how do you know where it occurred? And you know by using diagnostic levels of these hormones. So what I'm going to tell you is that it could be primary hyposecretion, secondary hyposecretion, or tertiary. So the way that you do it is you measure the levels of the hormones at each place. And you always start from where the symptoms occurred. So you're going to start from the bottom. So let me give you this example, and then I'll let you do the next one. If I had a primary hyposecretion of thyroid, what will happen is my level of T3 and T4 would be very low, right? But my level of TSH would be normal to actually slightly high, right? Because I'm not getting any feedback. Do you see? Okay. And my level of TRH would be what? Normal to high. Do you see that? Okay, so that would be a primary hyposecretion. The problem occurred with the thyroid. So now I want you to look at what would happen if the problem was actually with the anterior pituitary. So it's a secondary hyposecretion. So my level of T3 and T4 would still be low, right? But my level of TSH would be what? Low. And my level of TRH would be high. Exactly. Does that make sense? So you can tell where it's occurring by looking at the hormone level. Okay, so how about a tertiary hyposecretion? That means that the problem was not at the bottom, not second, but all the way up at the top, right? So a tertiary hyposecretion would have what levels of T3 and T4? Low. What levels of TSH? Low. What levels of TRH? Low. Do you see? Okay, now, so you would treat them differently if they were primary, secondary, or tertiary. Although, interestingly, they don't usually treat tertiary ones any different than secondary ones, because the problem is that you have to get this through the blood-brain barrier, right? So a lot of times they just lump the tertiary and secondary together. So why do they care? Well, with a situation like that, you want to know whether it's actually going on in the hypothalamus or the anterior pituitary, because you might be able to predict, for instance, some other disorders. If it was a hypothalamus tumor or a hypothalamus disorder, then you might end up with all different kinds of metabolic disorders. From my current level of knowledge, and I haven't checked over the last 2 years really, you can't even get tested for TRH levels unless you go to an endocrinologist. At your primary care or your OBGYN, they can test for T3, T4, and TSH, but they don't usually test for TRH, which is really low concentration anyway. Okay, so primary hyposecretion, remember, if it's primary, it occurs where the symptoms occur, right? Okay, so if it's a primary hyposecretion, it could be genetic. For instance, like type 1 diabetes, if you got it as a child, it's generally a genetic defect, right? You have a problem in some part of the biochemical pathway to, for instance, produce insulin, so you're not producing it normally, right? So a genetic defect in the gene for a hormone, or does it make sense to you that an iodine deficiency could cause a primary hyposecretion of thyroid hormones, T3 and T4? It wouldn't be secondary, would it? So if you had an iodine deficiency, guys, tell me about these levels of these hormones. What would T3 and T4 be? Low, because I didn't have any iodine. What would my level of TSH be? High, right? So a deficiency in some nutrient that you needed to make it would be considered a primary hyposecretion. So a dietary deficiency of a precursor to a hormone, iodine deficiency, can cause hyposecretion of what two hormones? T3 and T4, exactly. Or an autoimmune disease. Most autoimmune diseases are going to cause hyposecretions because they destroy the cells, right? So autoimmune diseases, we have a couple of these examples. Addison's disease, which we talked about before, that was the one that JFK had, right? And it is often called primary, as in primary hyposecretion, adrenal insufficiency. And it's actually a hyposecretion of two different hormones. Do you remember which ones those are? Cortisol and cortisol and aldosterone. So this isn't a glucocorticoid, but do you see why messing with your glucocorticoids will cause problems with aldosterone? Do you see? Because corticosterone is the precursor to aldosterone, right? So that's the connection between those. So this is primary hyposecretion of cortisol and aldosterone. Usually this is due to exposure. For some reason, this one they actually figured out. For some reason, exposure to the tuberculosis pathogens, whatever antibodies you produce in response to those like to attack your adrenal cortex. They think that JFK got exposed to TB when he was in the war, and then fast forward 10 or 15 years, 20 years, and then he ends up with Addison's disease, autoimmune disorder. Okay. Type 1 diabetes mellitus. This is hyposecretion of what hormone? Which hormone? Insulin, exactly. If it occurs in adulthood, it's more likely to be autoimmune, right? If it occurred in childhood, it was more likely to be genetic. Just depends on when it occurred. And then Hashimoto's disease. This is hyposecretion of what hormone? T3 and T4. Yeah, it's generally autoimmune. That make sense? Okay, so secondary and tertiary hyposecretions also exist, but remember that their symptoms will look just like it's primary. It's just that you figure out that they're secondary or tertiary by measuring your hormone levels. Okay, so those also exist. Okay, hypersecretion. We didn't learn as many hypersecretion disorders as we did hyposecretion disorders, but primary hypersecretions are often caused by tumors, either benign or malignant. It doesn't make any difference. A lot of times they manifest with secreting too much of the hormone. But we did have one example of an autoimmune hypersecretion. Do you remember which one that was? Autoimmune hypersecretion. No, that one's not autoimmune. Yeah, it's hyperthyroid. Do you remember that one? The weird example in which what happened is the antibodies that you produced in response to whatever pathogen are TSH agonists. Do you remember that one little weird example? Yeah, it was basically the one that ends up circumventing the system because the antibodies make your thyroid think it's being overstimulated over and over and over again. So that's the one example that you have of that. Okay, so let's talk about the hypersecretions that you know. Acromegaly is a hypersecretion of what hormone? Growth hormone in adulthood. And then pituitary gigantism is a hypersecretion of growth hormone in childhood, exactly. Okay. Secondary and tertiary hypersecretions also exist. You would just know from measuring the hormone levels. Okay. And then the responsiveness and hyper responsiveness. Okay. So these are disorders that you already know as well. So with a hyporesponsiveness disorder, there's nothing wrong initially with the hormone level. Okay. It's just that either the receptors are not binding to it or the target cell is not responding, right? So you're unresponsive to the hormone. But I want you to think about this. If you don't treat a hyporesponsiveness disorder over a long period of time, what do you think can happen to the level of the hormone? It goes up, exactly. So the deal here, and I'm not going to nitpick you or try to trick you on tests, but if you have a secretion problem and you don't treat it, you're going to end up with a responsiveness problem. If you have a responsiveness problem and you don't treat it, you're going to end up with a secretion problem, right? So you've got this sweet spot of time to treat endocrine disorders before you end up with another endocrine disorder, really. Okay, so hyporesponsiveness of target cells. So there's nothing wrong initially with the hormone secretion levels, but the target cells are not doing what they're supposed to do. Either they're unable to respond or they're not bonding to the hormone, or maybe the hormone wasn't actually metabolically active. Like if you release testosterone and don't metabolically activate it, then it's not going to cause a target cell response. But usually the way that people think about it is too few or insensitive receptors. That's generally what you're going to think. Okay, so you know this one. Type 2 diabetes mellitus is a hyporesponsiveness problem. It's a hyporesponsiveness of what kind of receptors? Insulin receptors. And it's often a result of what kind of regulation? Up or down? Down regulation, exactly. Make sense? Okay, so, and then testosterone insensitivity or androgen insensitivity is the one that we talked about that was a genetic disorder with your testosterone or androgen receptors. So this is a hyporesponsiveness of what receptors? Androgen or testosterone. That makes sense? That's the one where you have an XY fertilization event that ends up being an anatomic female, right? Because you're not responding to testosterone receptors. Okay, so with these, the hormone level starts out normal and then it will creep up as you are not responsive to it. Okay, and just to make this parallel, I'm going to teach you one hyperresponsiveness disorder. It's really hard to understand, and just so you know, I don't usually test over it, but I just hated not having hyperresponsiveness in there. It just offended my sense of like symmetry. So there is one, and it's hard for me to understand, so I don't think it's very easy for me to explain. Hyperresponsiveness of target cells. So thyroid hormone is a metabolic hormone. Do you agree? Okay. And epinephrine is also a metabolic hormone when it's released hormonally. So what happens is with chronic hyperthyroidism, it ends up upregulating your epinephrine receptors because they both have metabolic impacts on the cells. How does this manifest? If I've got a person with Graves' disease who doesn't treat it for a long period of time, in addition to having all of the other symptoms of Graves' disease, they're going to have this massive fight or flight response. Like a tiger walks into the room and their heart rate doesn't go up to 150, it goes up to 220 and it stays there for a while. They have this massive fight or flight response. It also tends to manifest in anxiety disorders and people with untreated hypersecretion of thyroid disorders, of a thyroid hormone. So basically metabolic impacts ends up impacting other metabolic hormones. This makes it really hard to treat if you let it get to the stage that you've upregulated your epi receptors. It also makes it really, really hard on the person because remember, epinephrine will actually cause mobilization of your resources into the bloodstream. And so you can end up with too much sugar and too much fatty acid in the bloodstream and you deplete your resources. So if you let it get to this point, it's a little harder to treat than it is in the early stages. But like I said, I don't usually ask about that one because it's hard to figure out how to phrase the question even. Okay, so the assignment is for every disorder that you learned, categorize it. Now, hypo or hyper secretion, hypo or hyperresponsiveness. Now, you can't categorize it as to primary, secondary, and tertiary unless, of course, I just give you levels of hormones, right? If I say, you've got this set of symptoms, weight gain, lethargy, exhaustion, and then these levels of T3, T4, TSH, and TRH, right? And then you could do both, right? You could say it's hyposecretion of, it's hypothyroid disorder, and then tell me whether it's primary, secondary, or tertiary if I give you enough information. Does that make sense? Okay. All right. Okay, so we're going to back up a little bit and just talk about receptors. This was all the way back in chapter 5, but people hate it, so I don't ever teach it when we're doing chapter 5 because now it's past the drop date, so you're stuck with me. So now we're going to do it. It's not very hard. It's just not very interesting. It's kind of irritating. Okay, so basically in order to get the target cell to respond, we're going to have to interact with the receptor, right? Okay, yes? Okay, so there are a bunch of different categories of receptors. We're going to learn 5 categories of receptors. Some of them you've already run into because we talked about receptors with neurotransmitters and that kind of stuff. Some of these you won't have seen before. But what's going to happen is in order for me, the hormone, to cause a response at the receptor, you're going to be the receptor. It depends on where the receptor is and what you're actually trying to accomplish. So if I'm a water-soluble hormone, I have to find a cell membrane receptor. Do you agree? So she's a cell membrane receptor. And if she's a cell membrane receptor, she's generally going to stay put in the cell membrane. Okay? But you guys, all of the rest of you guys, are part of the mechanics of the cell. And you, way back there, you are the nucleus. Okay? So what I wanted to do was cause protein synthesis, and I was a water-soluble chemical messenger. Do you see that I'm going to have to play a lot of telephone to actually get that to occur? Right? You guys play telephone as a kid? I tell you something, she turns around, tells her something, you tell her something. So with water-soluble chemical messengers, because I can't go inside the cell and she has to stay on the cell membrane, you're going to end up having a really long game of telephone before you can actually cause something to occur. That long game of telephone is called a signal transduction mechanism or a signal transduction pathway. It is how you get the target cell to respond. And sometimes it can be long if I'm a water-soluble chemical messenger. But what if I'm lipid-soluble? If I'm lipid-soluble, I can go straight past all of you guys, straight to the source, right? I can find my receptor all the way in the nucleus of the cell, right? So it's like a shorter game of telephone. Does that make sense? Okay, so these signal transduction pathways or signal transduction mechanisms, they depend on you knowing the chemistry of the hormone, right? You have to know if it's water-soluble or a lipid-soluble. So these signal transduction mechanisms or pathways. So let's do the simplest one first. The shortest game of telephone is with a lipid-soluble chemical messenger, yeah? And with a lipid-soluble chemical messenger, what I can do is, watch. I can go straight through the cell membrane without asking permission. I find the receptor either in the cytoplasm or it can even be all the way in the nucleus because remember the nuclear envelope is lipid too, right? So you can go straight to there. And then what is this going to do? So let's give you an example. This time I want you to imagine that I am testosterone and you guys are a skeletal muscle fiber. What testosterone does to skeletal muscle fibers is it causes protein synthesis of actin and myosin, right? That's why a lot of testosterone, of course, is going to build your muscles, right? So what I need to do is I need to go all the way to the nucleus and bind to the receptor, and then that new hormone receptor complex will go and bind to, for instance, the actin gene and cause transcription of the actin gene and then translation. And I skipped all of the telephone except for that last little part of it, right? So what it's going to do is it's going to go straight to the actin gene and cause transcription of actin, and then it's going to leave the nucleus to go to the ribosomes and cause translation of actin, right? So this process that is only possible with lipid-soluble chemical messengers is called gene activation, right? The receptor is all the way inside the cell. So this is just activating the specific gene. So I'll show you an animation for gene activation. So here is how gene activation works. So this one's going to use aldosterone.
Lipid-soluble hormone that can easily diffuse through the plasma membrane. Inside the cell, aldosterone binds with an aldosterone receptor molecule in the cytoplasm. The aldosterone receptor complex moves into the nucleus and binds to DNA. The binding of the aldosterone receptor complex to DNA stimulates the synthesis of messenger RNA, which codes for specific proteins. The messenger RNA moves from the nucleus into the cytoplasm and binds to ribosomes, where it directs the synthesis of specific proteins. These proteins produce the response of the cell to aldosterone.
So what is this protein? I don't know. It's something that didn't exist before, and therefore the cell is either structurally or functionally different than it was before, right? If it's a new enzyme, then you can behave differently. Or if it's a new structural protein like collagen, then your structure is different. The only thing that I can tell you is that that changes something about the cell. Otherwise, why bother, right? With this metabolically expensive process. Does that make sense? So this is called gene activation. This is the shortest game of telephone that you're going to play because the chemical messenger goes straight to the source. Okay. Now, if I'm going to use a water-soluble chemical messenger, then I am going to end up interacting with something that is limited to the outside of the cells. So there's a bunch of in-between steps. So with water-soluble chemical messengers, these only interact with plasma membrane receptors, right? It has to be on the plasma membrane. And there are 4 types of receptors that you're going to run into. Now, why bother talking about this stuff? I have to, not because it's interesting or fun, but because an awful lot, it's not, I don't even like teaching about it at all, but an awful lot of what you guys are going to do, for instance, if you went into pharmacy or pharmacology, does it make sense that you're going to spend a decent amount of time talking about receptors if you're in pharmacy or pharmacology? And so what I'm trying to do here is just to teach you the 5 major types of receptors that exist, okay? And one of them you just learned, the intracellular receptor that starts gene activation, is for lipid-soluble chemical messengers. And then there's a lot more water-soluble chemical messengers, right? So there's going to be 4 different types of receptors for those. One of them you've already met, because remember that, for instance, what type of receptor does acetylcholine interact with on skeletal muscle to cause contraction? The receptor is what? It's a nicotinic cholinergic receptor, but what does it do? When acetylcholine binds to that receptor, what does it do? It opens an ion channel, right? So one of the receptors that you already know is receptors that are also ion channels. Don't you already know that? Yeah? Okay, so let's just look at that. So we looked at a bunch of them when we were doing the nervous system. So here's a receptor, right? Chemical messenger binds to it, opens an ion channel, right? And what happens when you open the ion channel? Well, it depends on the cell. I mean, you could depolarize it or hyperpolarize it, right? Or you could, for instance, the presence of this new ion inside the cell could cause exocytosis or apoptosis or any number of things. It's just going to change the activity of the cell, right? That makes sense? So this is a really fast way of changing the activity of the cell. We're going to look at all of the other types of receptors, but this one, moving ions in and out, especially if it's ions that with an electrochemical gradient that favors them moving in and out, can be a really fast way to change what's happening with the cell. So this is a receptor that is also an ion channel, right? And we've met a bunch of those, so you guys are not going to be. So what happens inside the cell is specific to the cell, right? But something's going to happen inside the cell. Okay, so the next type of receptor we haven't talked about before, and it's a receptor that is also an enzyme. Okay, so let's just look at this one. It's not really too bad. Okay, so here's your receptor, right? So what I have is a single string of amino acids, but one end behaves as a receptor and the other end behaves as an enzyme. Okay, but it's the same string of amino acids and it's stuck in the cell membrane. So what's going to happen when the chemical messenger binds? Chemical messenger binds, and what it does is activates the enzyme. The enzyme was inactive before. This category of receptors that are also enzymes are called the tyrosine kinase receptors, because what they do is what a kinase does. What's a kinase do? What's a kinase do? Yeah, it phosphorylates something, right? It takes a phosphate off of ATP and it slaps it onto something else. Why are these called tyrosine kinases? Because they specifically phosphorylate the tyrosine amino acid on another protein. Who the hell cares? Well, that's a really common way of activating a protein, right? That makes sense? So I had an inactive protein. When something binds to the receptor, I activate it. And now the cell behaves differently than it did before. So what might it do? Oh, I don't know, difference in metabolism, difference in cell division, difference in protein synthesis. I have an active enzyme that I didn't have before. So now I can behave differently. Does that make sense? So these are called the tyrosine kinase receptors. Okay, and there are a whole bunch of these. These are obviously not going to be as fast as an ion receptor because phosphorylation is not, forming covalent bonds is not as fast as just like diffusing ions in. Is that okay? Okay, so these are called tyrosine kinase receptors. Now, the next category is almost like these, but it's got a difference. Do you see how this one is all one string of amino acids, right? So the receptor and the enzyme are all the same string of amino acids. With the next one, these are called the JAK kinases. The receptor end and the enzyme end are two different proteins, but they're stuck together. So your textbook does not have a picture, so I pulled one from a different textbook for you. Okay, so let me just show you how they recept. Okay, so this is the one that we just looked at. The receptor is also attached to the tyrosine kinase, just so you can sort of orient. And notice that it's all the same blue protein. Okay, these are called the JAK kinases. The receptor is a different string of amino acids than the other one. But what does it do? Pretty much the same thing. It's going to do kinase activity and therefore activate a protein. So these are called the JAK kinases, and the reason I'm having you learn these is because these are the most common category of receptors that exist in the body. There's so damn many of them that they got tired of naming them, and literally they are JAK kinases because JAK stands for just another kinase. There were so damn many of them that they got tired of naming them. They used to like name it after your favorite scientist or whatever, or name it after yourself. And then now in biochemistry labs, where they're like, I found a kinase. And they're like, oh, did you also find hay in the barn? Because honestly, you could throw something and hit a kinase. So these are like super duper common, JAK kinases. Okay, so those produce a similar cellular response. You have a newly active enzyme that you didn't have before. Okay, so the last category are the second most common that you ever run into. JAK kinases are the most common, and then these are the second most common. And these are called G protein receptors. And I already kind of introduced you to these just a little bit. The G protein receptors are like the most complex. I don't even know how the hell these things evolved. They are the most inefficient way of accomplishing anything. These are the G protein receptors. In Texas, we would call this going around your ass to get to your elbow because they like involve 5 different steps where you could have done one. So here's how this occurs. So the chemical messenger binds to the receptor, but there are 3 proteins associated with that receptor that are actually not part of the receptor. They're just associated with it. They are all run by guanosine triphosphate, GTP instead of ATP. That's why they're called G protein receptors. And what's going to happen is a really complex combo shot and pool. Here's your pool cue right here, the chemical messenger. Here is the cue ball right here, this receptor. But what's going to happen is I'm really trying to hit that pocket. But I'm going to do it by actually going through all 3 of these balls. So what's going to happen is you're going to bind here. It's going to change the shape of the receptor. It's going to slide these through the cell membrane to smash into that protein, which is what you were trying to manipulate. If it was an ion channel, you were trying to open it. If it was an enzyme, you were trying to activate it. Okay, but you did it the most complex way possible. So I'm going to show you an animation. We'll show you how this works. So imagine that this is part of this story, okay? Because they didn't draw one in there. Okay. All right, so here we go. We have a water-soluble chemical messenger. It's going to bind to the receptor. And then notice I'm going to use GTP to get these things sliding through the cell membrane. And they're going to slide through.
Alpha subunit
And
of
then
the G.
the alpha subunit is going to slide off of the other 2. And then the alpha subunit is going to smash into that 4th protein to open or close that protein. These are super duper common. And it's just like amazing that they ever work at all. really. And this is how a lot of neurotransmitters work. Does it make sense to you that these would not be the fastest target cell response? These are the slowest target cell response. And then they're going to go all the way back into position so that you can use them the next time. So notice I'm going all the way back into position that you can use them the next time. So literally this has 1, 2, 3, 4, 5 proteins where one of them would have done, right? Okay, but apparently they work because we've got a whole bunch of them. A bunch of our neurotransmitters work with G protein receptors. Not the fastest neurotransmitters though, not surprisingly. So those are, so we're going to back up just a little bit to make sure that we tell this story so that it makes sense. So if I am a lipid-soluble chemical messenger, then how am I going to cause my target cell response? You go straight in and you do what process? What's it called? Gene activation, right? But if I am a water-soluble chemical messenger, I have 4 possibilities for types of receptors that I'm going to interact with. If, for instance, I were acetylcholine and you guys were a skeletal muscle, I will interact with a receptor that is also what? Ion channel, exactly. Receptor that is also an ion channel. Okay. Or I can interact with the receptor that is also an enzyme. What's that category called? Tyrosine kinases. Or I could interact with a receptor that is bound to an enzyme, and that is called what? JAK kinases. Or I can involve my 4 closest friends just to try to get a single job done, and those are called what? G protein receptors. There you go. So that's not anybody's favorite thing to study, and because it's not, it's very commonly missed on the endocrine test. So go ahead and study them. I know you don't like them. I don't like them either. But you have to. So what is the target cell going to do at the end of all of this rigmarole? Something different than it did before is all I can tell you. New enzyme activity, new metabolic activity, new structural protein, cell division, change in metabolism, something that it didn't do before. Okay? All right. So the last part in this set of notes is simply making sure that you know the feedback mechanisms. So students very often think that they know these, but until you put it together, you might not. So what I'm asking is, in each of these situations, do you actually know what is actually feeding back in the loop to tell you when to start or stop secreting the hormone? So there are basically 3 ways that you control hormone secretion. You control it by a hormone feeding back, right? And you should know countless examples of that. But sometimes it's not a hormone that feeds back. It's a nutrient or an ion that feeds back through the bloodstream. Can you think of any situation in which it's a nutrient or an ion that feeds back? Calcitonin and calcium. Yes, exactly. So do you see it's not the calcitonin that's feeding back to tell you when to stop secreting calcitonin, right? It is calcium that's feeding back. And then the third one, and the hardest pictures to draw, but not hard to understand conceptually, is the nervous system is going to be the feedback mechanism. Can you think of any situation in which the nervous system tells you when to start secreting a hormone and when to stop secreting a hormone? Can you think of any situation in which it's the nervous system? Yeah, it's epinephrine secretion with fight or flight, right? Isn't that in the nervous system? Tiger! Oh, not a tiger, a dude in a tiger suit, right? It's the nervous system. Or how about oxytocin, right? Oxytocin is secreted because of nipple stimulation, right? It causes milk letdown. Does that make sense? Okay, good? Okay, all right, so what we're going to do is with your hormones that you learned, most of them you're going to be able to categorize as one of these 3 types of feedback loops. So because of course your hormones are not secreted in constant quantities. So you change the level depending on the body's needs. Otherwise this whole story would have been way, way simpler. So it's generally negative feedback inhibition, but the thing that I'm asking you to make sure that you understand is what is feeding back. Okay, what is the feedback? Okay, so there are 3 types of input. They call these a non-hormone substance feeding back. They call that humoral control. But not because it's funny, but because remember, they used to refer to the body fluids as humors. Vitreous humor, aqueous humor, Yeah, so that's a really old term. And then there's hormones feeding back, that's called hormonal control. And then there's nervous system feeding back, and that's called neural control. So we'll see examples of each one of these. Okay, so let's just categorize some things that you already know. Control of hormone secretion by ions or nutrients feeding back. Okay, this is humoral control. The classic example is, of course, insulin. What controls whether you secrete insulin or not? It's blood glucose levels, right? So if you don't know this, you sure as hell should, right? So insulin is pretty damn important. So if your blood glucose is high, then the beta cells of the pancreas secrete insulin, and then the cells uptake insulin, and that drops your blood glucose level. But the important thing is what's feeding back right here is the glucose level, not the insulin level, right? Couldn't you tell this same story with glucagon, just changing the levels, right? So insulin and glucagon are both regulated by the glucose feedback, right? Okay, all right, so Haley said calcitonin. You guys know that story too, right? Blood calcium levels. It's blood calcium levels that feed back to tell you when to start and stop secreting calcitonin and PTH, is it not? So that's another example of a non-hormone substance controlling hormone secretion. And then this one you might not know, but I want you to look at it and see if it fits into this category. Aldosterone is kind of a complicated hormone, and its feedback mechanism is, well, it responds to sodium levels and potassium levels, and also sometimes blood pressure. But I just want to show you one of the ways that you control aldosterone secretion. And I'm not going to say memorize this, but I want you to see if it fits into this category, if it makes sense that it goes in this category, okay? So it's this one right here, okay? This is one of the ways that you control aldosterone secretion. Your blood potassium goes up, your adrenal cortex responds by secreting aldosterone, and then your kidneys increase the amount of potassium you pee out, your potassium excretion, which drops your potassium level, and that's what feeds back to tell you when to stop secreting aldosterone. Do you see that this fits into the category that we've been talking about? Because it's not aldosterone level that's feeding back, it's an ion or nutrient, okay? So not did you know that one before, but do you see that it goes here, right? Okay, all right. All right, so that's potassium levels and aldosterone. All right, so the next category is control of hormone secretion by other hormones. And you should be able to think of freaking countless examples of control of hormone secretion by other hormones. Because isn't almost every single hypothalamus, anterior pituitary, third endocrine organ hormonal feedback? Isn't it? Like whether you're talking about thyroid, right? That's a classic example of hormone feedback, because what is it that feeds back? It's T3 and T4, right? Or cortisol feeds back, for instance. So there are countless examples of that. So control of hormone secretion by other hormone. It generally involves hypothalamus, anterior pituitary to third endocrine organ. So the general pattern is a 3-hormone sequence: hypophysiotropic, other tropic, and then third hormone. So classic examples, if you can't do anything else on the test, make sure that you can do this one, right? The thyroid, okay? So this is a classic hormone feeding back loop. And then the other classic examples are, well, we just did it there, right? Your sex hormone secretions. If you just concentrate on this half, right? If you just concentrate on that half, it's a really classic, easy example. Doesn't matter whether it's estrogen or testosterone, either one of them works for that part of the story. Cortisol is another one. And cortisol is another one, especially if you take stress out of the equation. If you take stress out of the equation, what is it that actually feeds back? It's cortisol that feeds back. So it's hormone controlling hormone secretion. That makes sense? Okay. So we've got testosterone and estrogen and cortisol and all of that kind of stuff. So last thing today, and then we'll talk about nervous system next time, because I don't want you to get too much ahead of the other class. So obviously we are seeing that the hypothalamus is in control of an awful lot, right? But to back up one step and say, okay, if the hypothalamus is controlling so much, then what is controlling the hypothalamus? And the real interesting answer is that the hypothalamus is really the crossroads between the nervous system and the endocrine system, because the hypothalamus is taking in sensory information about what's going on, right? Like day-night cycling, it's like assessing light and temperature to figure out timing. It's also assessing metabolic information, but then it's also, I mean, look at all the receptors that the hypothalamus must have for hormones, right? So it's both receiving a bunch of sensory information and receiving a bunch of hormonal information. And then what the hypothalamus does is it just sort of synthesizes all of that collectively to figure out subconsciously when to secrete hormones and when to stimulate the nervous system. This is like the crossroads of those two body systems, the nervous system and the endocrine system. So the question is, since the hypothalamus seems to control many functions, what controls the hypothalamus? Nervous system input, light, temperature, metabolism, circadian rhythms, and then hormonal feedback. Every single hormone that feeds back to the hypothalamus, there must be receptors on the hypothalamus for that hormone, right? So you can imagine how that little bitty freaking nugget-sized region of your brain has massive nervous system and endocrine system function. Just that little one example right there. So it has tons and tons and tons of receptors. Okay, we're going to stop there for today, and we'll do nervous system next time. Next time we're going to talk about the acute and chronic stress response, which is one of my favorites. So really, really interesting stuff. Some new research that's been going on over the last 5 years. And your quiz is not until a week from today. So you're going to have it on Monday instead of Wednesday.